Our brain is the body's control center, protected by a highly selective shield called the blood-brain barrier, or the BBB, which only lets essential molecules in while blocking harmful substances. But this impressive defense comes with a downside. The BBB also keeps out many drugs needed to treat brain diseases like Alzheimer's and Parkinson's, making treatment delivery to the brain a challenge. One solution that scientists are exploring is nanoparticles. The surface of these particles can be customized with ligands to attach to specific receptors on the BBB's endothelial cells, allowing them to cross through a process called transcytosis. However, studying the function of these nanoparticles poses a problem, as animal models often fail to accurately predict human responses due to their significantly different physiology. That's where a fascinating new tool comes in: a blood-brain barrier on a chip. And no, we're not talking about some electronic chip for a digital simulation of the BBB. No, we're talking about a physical model built with living cells and tiny channels to mimic the actual blood-brain barrier environment. In a 2020 study, scientists indeed designed such a chip. The chip contains two key compartments separated by a porous membrane: a lower brain channel with parasites and a 3D astrocyte network, and an upper vascular channel lined with endothelial cells. One exciting finding involves a nanoparticle called EHNPA1, which mimics HDL, or the good cholesterol. The researchers hypothesized it could cross the blood-brain barrier by attaching to SRB1 receptors on endothelial cells. In one key experiment, the researchers fluorescently tagged the nanoparticles and quantified the percent that remained in the vascular channel after its administration. In the control group, with normally functioning receptors, only 25% of nanoparticles remained in the vascular channel. Meanwhile, when these transcytosis receptors were blocked by BLT1, nearly double the amount of nanoparticles remained in the vascular channel, confirming that the SRB1 receptor is key for crossing the blood-brain barrier. This discovery is exciting because it means that the HDL-like nanoparticles could be a potential delivery system for brain-targeted drugs. Of course, this isn't a perfect replica of the human brain environment. Critics argue that it oversimplifies the complex brain-vascular interactions. An article even reports that the proposed compartment arrangement does not allow for the simultaneous visualization of all involved cell types, thereby hindering comprehensive microscopic analysis. But this model is still a major step forward. Instead of being discouraged by its limitations, each version builds on the last, helping us unlock more secrets of the brain, one step at a time.